Good morning, my dear students. Welcome to our zoology class. I hope you are very happy and you are studying regularly. And many students sending the answer to me. I really appreciate the students, those who are sending the answer to me through the WhatsApp. So regularly study all subjects. Okay. Listen. Already we have completed the first three chapters. Okay. So today we are going to start the next chapter, fourth chapter. Today our lesson name is Principle of Inheritance and Variations. Principle of Inheritance and Variations. Listen students, genetics. I hope you all hear this word genetics. So here genetics means it is a branch of science or it's a branch of biology and deals with the inheritance and variations. Okay, inheritance and variations. So this branch, okay, this genetic branch explain how the characters are transferred from parents to the offspring and what is the reason for the transformation of characters from one generation to another generation. Okay, so maximum this branch of this biology genetics explaining how the characters are transferred from one generation to next generation. Okay, so it's completely explaining about the genetics. Understand? Genes. So genes are the one of the important factors present in all the cells. And these genes only responsible for some hereditary characters. Hereditary characters means in Tamil we can say பாரம்பரிய குணங்கள். Okay, so the hereditary characters are transferred from one generation to next generation because of the presence of these genes. So genes are the very important factors playing a very important role in the hereditary process. Understand, dear students? So in this lesson we are going to study about many topics related to this genetics. Okay, for example, multiple allelism, sex determination in different living organisms, sex linked disorders. Okay, like that different types of topics we are going to study in this lesson. So today we are going to study about only one topic. The topic name is multiple allele. Okay, multiple allele. Say here multiple allele meaning three or more alleles. Okay, three or more alleles located in the same homologous chromosome, but they are controlling only one particular character. So these type of alleles are called multiple alleles, and this type of hereditary process is called multiple allelism. So once again repeat the meaning of this word multiple allele. Three or more alleles, three or more than three alleles togetherly controlling a particular character, controlling a single character, and all these alleles are located in a single homologous chromosome. Understand? So this condition is called multiple allele, and this type of hereditary process is called multiple allelism. One of the best example for this multiple allelism is human blood group. So human blood group is one of the example for this multiple allelism, because the human blood group, okay, blood group of the human being is determined by three alleles. Okay, three alleles. Okay, say chemically the blood, look at this, chemical structure of the blood is different from one person to another person. Okay, chemically the structure of the blood is different from one person to another person, and the reason for this differences is presence of one antigen. That means you all know our blood has many cells, RBC, WBC, platelets, like that. Different types of cells are present in the RBCs. RBCs are otherwise called erythrocytes. So the outer surface of the RBC having one antigen. So one antigen is present in the on the outer surface of the antigen. Understand? So, these antigens are classified into two types, antigen A and antigen B. So, first these two antigens are discovered by one scientist. His name is Karl Landsteiner. Okay, Karl Landsteiner only first discovered the surface antigen, antigen A and antigen B. So, these two antigens are present in the outer surface of the RBCs of the human blood. Understand? And these two antigen, antigen A and antigen B, okay, these two antigen was first discovered by one scientist. His name is Karl Landsteiner. Okay, Karl Landsteiner. Then, and already I told you, so the blood group of the human being is determined by these antigens. Okay, in some persons RBCs having A antigen, some persons RBCs having B antigen, and in some antigen, okay, some person's body these two antigens are absent. So based on the presence or absence of these two antigens, the human blood group is divided into three types. Okay, A group, Group A, Group B and Group O. Okay, so Karl Landsteiner only first discovered these two antigen, antigen A and antigen B. So based on the presence or absence of these two antigens, the human blood group is divided into three major groups: A group, B group and O group. Understand all of you? Then another two scientists. The scientist name is Van de Castell. First scientist name is Van de Castell. Then second one name is Sturley. Sturley. So these two scientists, these two scientists are the students of this Karl Landsteiner. These two scientists are the students of this Karl Landsteiner. So these two scientists togetherly discovered one more blood group in the year of 1902. Name of the blood group is called AB blood group. So totally three scientists togetherly discovered four blood groups. Blood group A group, B group, O group and AB group. Here this AB group only discovered by these two scientists, Van de Castell and Sturley. These two people only togetherly discovered this AB blood group. I hope you all understand these important points. So Karl Landsteiner discovered three blood groups: blood group A group, B group and O group, based on the presence or absence of the surface antigen present in the RBCs. Then his students name Van de Castell and Sturley. These two people together discovered one more blood group. The blood group name is AB, and they discovered this AB blood group in the year of 1902. Understand all of you? Then the next point. See, one scientist. His name is Bernstein. See, in the year of 1925. In the year of 1925, one more scientist, his name is Bernstein. This person only first discovered that the blood group of the human being is determined by genes. So the genes only responsible for the blood group of the human beings. Understand? And the name of that gene is called L gene or I gene. L gene or I gene. So the genes responsible for our blood group is called L gene or I gene. Understand all of you? And these genes are, this L gene or this I gene is located in the 9th chromosome of our somatic cell. That means body cells. So in our body cell, in the 9th chromosome, one gene is there. Name of that gene is called L gene or I gene. This gene only responsible or determining our blood group. Understand all of you? Then in the beginning I told you, when I start the lesson I told you, the blood group of the human being is determined by three alleles. Okay, so here the allele's name is IA. Here these genes. Okay, IA, IB, and IO. These three alleles only determining or responsible for our blood group. Okay, dear students, I hope you all understood this point. Once again, I repeat this point. In the year of 1925, Bernstein, okay, this person only first discovered that our blood group is determined by one gene. Name of that gene is called L gene or I gene, and this gene is located in the 9th chromosome of our body cell. Then this gene consists of, okay, this gene has three important alleles. Name of that alleles is called IA allele, IB allele, and IO alleles. Understand all of you? Understand all of you? Then here this IA and IB alleles are always dominant than these IO alleles. Okay, IA allele and this IB allele is dominant than this IO alleles, because these two alleles, IA allele and IB allele, producing some enzymes, but this IO allele will not produce any enzyme. So this allele is called null alleles. This allele is called null allele. So I hope you understood this point. So the I gene or the L gene having three important alleles. They are IA allele, IB allele, and IO allele. Here these two alleles are always dominant than this IO allele because they are secreting the enzyme. This allele will not secrete any enzyme, so this allele is called null allele. Allele is called null allele. So if these alleles, okay, present in the RBCs, okay, so these alleles only determining, okay, these alleles only determining the blood group. For example, this IA allele is determining the A antigen. Already I told you two surface antigens are present in the cells. So IA allele determining the A antigen, IB allele determining the B antigen, and this IO allele will not determine any antigen. So in the beginning I told you two antigen, A antigen and B antigens are present in the outer surface of RBC. Here this A antigen is produced by this A allele, and the formation of this B antigen responsible B alleles, IB alleles. Understand a few? I hope you all understood this point. Okay, then come to the next point. Say a child receive one allele, okay, one allele from each parent. From each parent, listen very carefully. See, I, A, I, B, I, O. This is the alleles of father. Then IA, IB, IO. This is the alleles of mother. Okay, so already I told you 9th chromosome of all the body cells having these three alleles. So in father's 9th chromosome these three alleles are there. In mother's chromosome 9th chromosome also these three alleles are there. So when we draw the phenotype, we will get 6 phenotypes. We will get 6 phenotypes. Okay, and 4 blood groups. Listen very carefully. So this allele and this allele. So when a child receive IgA allele from the parent and IgA allele from the mother, so IA. Then from father IB, mother also IB. From the father IO, mother also IO. Now we got 3 phenotypes. So here one IA, from father IA, mother also IA. So this is one phenotype. Then from the father IB, from the mother also IB. Another one phenotype. Then from the father IO, mother also IO. So the third phenotype. Okay. Then next, sometime the child may receive. Okay, the child can receive this IA from the father. Okay, listen very carefully. IA from the father from the mother I, B, then from the father I, B, then from the mother I, O. Okay, now we got two phenotypes. Understand a few? Then from the father I, O, then from the mother I, A. Now we got six phenotypes. This is one phenotype, second, third, fourth, fifth, sixth phenotypes. Understand? Six phenotypes are there. Understand a few? So the blood group totally here IA determining A blood group, IB determining B group blood group, then IO determining O blood group, then IB determining AB blood group. These two phenotypes will not determine any blood group, because here one gene is what gene, one allele is what a null allele. Here also one allele is null allele. Understand a few? So when we already told you, a child receive one allele from each parent, from mother also, from the father one allele, from the mother also one allele. So finally when we draw this phenotypic character, we will get six phenotype. Okay, understand a few? Hope you understood. So this is very important 5-mark question. Blood group determining in human beings. Understand a few? Then, so once again repeats. Okay, see this human blood group. Okay, the blood group, the chemical nature of the blood group is different from one person to another person, and the reason for the differences is what is the presence of one surface antigen present on the outer surface of the RBCs. Understand? And Carl Landsteiner identified or discovered two surface antigen. One antigen name is called A antigen, the second antigen name is called B antigen. So based on the presence or absence of these two antigens, the blood group, he discovered three types of blood group. Blood group A, blood group B, and blood group O. Then two more scientists, Van de Castile and Story. These two people are the students of this Karl Landsteiner, and they discovered one more blood group. That blood group name is AB blood group, and they discovered this AB blood group in the year of 1902. Understand a few? Then this Bernstein, in the year of 1925, Bernstein only first discovered the blood group determining in the human beings is because of the presence of genes. So genes only responsible for the blood group. Understand? And these genes located on the 9th chromosome, and name of the gene is called L gene or I gene. And this L gene or I gene having three alleles, A allele, IB allele, IO alleles. Understand? Here this IA allele and IB alleles are always dominant than this IO allele. Understand a few IO alleles. Understand? Then, so when a child receive, okay, so a child receive one allele from the father and one allele from the mother. So when we draw this phenotypic character, we will get 6 phenotypes and 4 blood groups. Understand? Understand a few? Then see, I have one more point here. This already told you these two alleles are dominant than this IO allele. Because the reason is I said the reason these two alleles secreting enzyme. Okay, producing a enzyme. For example, this IA allele, okay, IA allele producing one enzyme. Name of that enzyme is called N-acetyl galactose transferase. N-acetyl galactose transferase is the enzyme produced by this IA allele. Understand a few? Then this IA allele can add one more enzyme. Name of that enzyme is called N-acetyl galactosamine. Okay, N-acetyl galactosamine is the enzyme added by this IA antigen. Sorry, IA allele. Understand a few? Then this IB allele produces a enzyme. Name of that enzyme is called galactose transferase. Okay, galactose transferase. And it can add one more enzyme. Name of that enzyme is called galactase. I hope you understood this point. IA alleles producing one enzyme and adding one enzyme. This IB allele also producing one enzyme and adding one enzyme. Here IA allele producing what enzyme? N-acetyl galactose transferase. And this allele adding N-acetyl galactose. Okay, galactosamine. Then this IB allele producing galactose transferase, and this allele and galactose. Understand? And this IO allele will not produce any enzyme. Already told you. So this allele is called null allele. This allele is called null allele. Understand a few? Then next, say the RH factor. See, RH factor. This is one of the important factor present in the blood of the human beings. Normally, this RH factor was first discovered in one monkey's blood. Name of the monkey is called rhesus monkeys. Okay. So, in rhesus monkeys blood only, this RH factor was first discovered by two scientists, Karl Landsteiner and Alexander Weiner. So, Karl Landsteiner and Alexander Weiner only first discovered this RH factor in the blood of rhesus monkey in the year of 1940. In the year of 1940. And later this factor was discovered in the human blood also. I hope you all understood these points. Okay. So, first this RH factor was identified in the blood of the rhesus monkey. Then later it was identified in the blood of the human beings also. Understand a few? Understand? Here, this RH factor is classified into two types, RH positive and RH negative. RH positive and RH negative. And this RH factor is due to the presence of one antibody. Name of that antibody is called D antibody. Name of that antibody is called D antibody. Sorry, D antigen. So, if a person's blood having D antigen, name of that blood is called, name of that RH factor is RH positive factor. If there is no D antigen, that blood is called RH negative blood. That blood is called RH negative blood. Understand? I hope you understand this point. Then, normally, these antibodies, okay, antibodies, RH antibodies are not present in any person's blood. But at the same time, when a RH negative person, okay, RH negative person receiving the blood from the RH positive person, okay, RH negative person receiving the blood from the RH positive person, it is not danger to anybody. But at the same time, when a RH positive person receiving the blood from the RH negative person, it is very danger. I hope you all understood this RH factor. So, RH factor was first discovered by Karl Landsteiner and Alexander Weiner in the year of 1940. The year of 1940. And this factor is based on the presence or absence of one antigen. Name of that antigen is called D antigen. Okay. If this D antigen is present, that blood is called RH positive blood. If the D antigen is absent, that blood is called RH negative blood. Understand a few? So I hope you all understood this point. Then we are going to study about two important hypotheses. The first hypothesis is called Fisher-Race hypothesis. Fisher-Race hypothesis. See, these two people togetherly said there are three alleles are responsible for this RH factor. Three alleles are responsible for this RH factor. And all these three alleles are present in different loci of a homologous chromosome pair. Okay. So just to imagine this is a homologous chromosome. This is a pair of homologous chromosome. Three loci are there. This is one, second and third. Here also first loci, second loci and third loci. Okay, so already I told you three alleles. Okay, three alleles only determining our RH factor. This is said by these two people. So that already this hypothesis is called Fisher-Race hypothesis. See, this allele capital C or small c. Capital C or small c. Then capital D or small d. Then the next allele is capital E or small e. Here also capital C or small c, capital D or small d, and capital E or small e. Understand all of you? So this is a pair of homologous chromosome. Each chromosome has 3 locations or 3 loci, and in each loci 2 alleles are there. Okay, 2 alleles are there. So when we draw the phenotypic character, we will get many phenotype. For example, see, just imagine first this from this loci capital C, from this loci capital D, from this loci capital E. Then from this loci small c, from this loci small d, then from this loci small c. Okay, capital C, then capital D, this capital D, then capital E. Then this from this loci small c, small d, then small. So this is one phenotype. This is one phenotype. Then sometime from this side small c, small d, small e. Then from here also small c, small d, small e. Understand? Like that we can draw many phenotypic characters. Understand? So here what are the phenotypic character carrying this capital D? That phenotypic character is Rh positive phenotype. What are the phenotypes are carrying the small d, 2 small d's? They are called Rh negative blood, or they are called Rh negative phenotype. So listen very carefully. This Fisher and Race, these two people togetherly explain that this Rh factor determination is because of 3 alleles. And these 3 alleles are located in 3 different locations of a single homologous chromosomes. Okay. Here the alleles may be capital C or small c, capital D or small d, or capital E or small d. Understand? So from this, suppose in this chromosome these capital D's are, we are taking this capital C, capital D, capital E. From this side small c, small d, small e. So this is one phenotype character. Then from this side, from suppose in this chromosomes small c, small d, small e. In this chromosome also small c, small p, small e. So this is the next phenotype. Like that we can draw many characters, many phenotypes. So here what is the point is, here the point is, if a phenotypic character is carrying capital D, that phenotype is called Rh positive phenotype. If a phenotype is carrying 2 small d's, that phenotype is called Rh negative phenotype. That's all. I hope you understood this Fisher Race hypothesis is a very important 3-mark question. Then next one hypothesis. Next hypothesis name is Weiner hypothesis. Okay, so here this Fisher and Race said 3 alleles determining the Rh factor. Here this person Weiner saying totally 8 alleles determining the Rh factor. Totally 8 alleles determining the Rh factor. See, alleles names are capital R1, capital R2, then capital R0, then capital R Z, then small r, small r1, small r2, then small r y. So these are the 8 alleles. All these alleles are responsible for this Rh factor. Okay, all these alleles are responsible for Rh factor. Then, so when we draw this phenotypic character by using these alleles, we will get many phenotypes. We will get many phenotypes. What are the phenotypes carrying this capital R? That phenotypes are called Rh positive phenotypes. Then what are the phenotypes carrying 2 small r? They are called Rh negative phenotypes. Rh negative phenotypes. So I hope you understood these 2 hypotheses. This is Fisher-Race hypothesis, and this Fisher-Race hypothesis saying 3 important alleles determining the Rh factor, and this Weiner hypothesis saying only 8 alleles. Okay, 8 alleles determining the RH fact. Understand all of you? Understand all of you? Okay, my dear students, I hope you all understood these two hypotheses. Understand all of you? So, today's many 1 mark questions are there. Okay, we have studied many 1 mark questions. Then we studied one 5 mark question, ABO blood grouping. Okay, it's a very important 5 mark question. Then these two hypotheses are very important 3 mark questions. Very important 3 mark questions. So all study today itself. You all try to complete these question answers. Okay, so it's very important question from the 4th lesson. Okay, then after studying, send the answer to my WhatsApp number. Okay, if you put any mistake, I will rectify the mistake. Okay, so study well. We will meet in the next class. Thank you.